insights into the anti-cancer model of mode of action of Britannia somnifera.
So thank you, ma'am, for introducing me and a very good point to all of you. So I'm Dr. IL38 Kumar from Central University of Punjab. I'm working as assistant professor in Department of Biochemistry for the last 7 years. So I will be talking you invite two lectures that was related to pharmaceuticals. Now I'm dealing with nutraceuticals. So I will be talking on a much renounced plant that is known as Withenia somnifera, and that plant is being used in different medicinal systems across the world. So here I just put the representation of the particular plant that is Withenia somnifera. It belongs to Solanaceae, and we know about the pharmacy or you can say the pharmaceutical activity of this particular plant, and we heard about this plant in newspapers and in other, you can say, different literatures. But the PubMed indexed articles here, I just categorize those, and it shows that near about the 25% of the publications belongs to, you can say, the cancer. And out of that particular 25% of the literature is related to breast cancer, and that it reflects the importance of this particular plant in the field of breast cancer. So here again, this is a slide which just introduces you that the active, or you can say the active clinical trials are there on this particular plant or in product. So it reflects the importance of this plant in this advanced research. And now these are some methanoids, and these are the active anti-cancer compounds present in this particular plant. And I will be talking about this Vithaferin A. This is the pharmacologically active ingredient of this plant, and so many pharmaceutical, you can say, the activity are reported for this particular compound, and the various mechanisms have been deduced at molecular level and biochemical level. And this particular slide shows and indicates the beauty of the natural products. You can see here there are so many mechanisms of actions of a single compound, and that is of the, you can say, the natural origin. And here you can see the each and every cancer. There are so many cancers that this particular single compound or of natural origin is targeting so many cancers. And this is the beauty of the natural compounds when deal with the natural origin of compounds. Okay, so they are multi-targeted. They used to target so many signaling pathways, they used to target so many biochemical aspects of any organism. And the thing that is involved between that, the principle that lies between this particular beauty, I think that is the plant and the animals, we co-evolved. We co-evolved, and this is the thing how, and this might be the reason behind this particular thing, that a single compound is used to target multi-targets. Now this is the very basic slide that relates to the microRNA, and this is what these are the small non-nucleotide sequences that are regulatory in nature. And the occurrence of these miRNAs, or the percent of these miRNAs I just mentioned here, they are what they are the regulatory in nature, and the most important thing, they are related to their dysregulation is associated with the occurrence of a particular disease, or the severity of a particular disease. And this is the fact that initiates, or you can say that opens up a venue to a new era of, you can say, the therapeutic targets. So again, this is a small table that indicates that yes, miRNAs may act as therapeutics because some of the clinical trials have been done. And in the previous slide I mentioned the IL38 that has been approved for in phase 1 clinical trial. So I will be talking about the triple negative breast cancer. So breast cancer, you heard about the breast cancer, but the triple negative breast cancer, why triple negative breast cancer? Because it's a subtype of breast cancer, and it's a very dangerous thing, or you can say cancer, because these are the statistics you can find in the figures, and in newspapers you can find it. And here you can see that in India, the triple negative breast cancer, among other subtypes of the cancer, the prevalence is high. And that the second figure belongs to the occurrence of this particular disease among the various cities of our country. And the most important thing here, you can see the hormone-based breast cancer, that the first type of breast cancer, and HER2 positive, and then third is the triple negative breast cancer. Here you can see the incidence of the triple negative breast cancer, And the thing that made it worsens, it is the absence of the pharmacologically active targets here, and as well as the relapse of the disease, the aggressiveness of the disease, as well as the despite there you have got the very less chemotherapeutic ability in different. Here is it is what this is what the available treatment strategies in different. You can say the chemotherapy is there, hormone therapy, radiotherapy, and immunotherapy. These are the various strategies to manage this particular disease. And this is here I just point out some of the problems in the management of triple negative breast cancer, therapy failure, drug resistance, relapse, and metastasis. So these are the very crucial parameters which are related to the mismanagement of disease, or you can say in other terms we are not able to fully manage the disease. And one more critical point is that this particular triple negative breast cancer subtype used to occur in very younger women, and it is very alarming. This situation is very alarming. And there I just point out some of the miRNAs that have been studied for the management of the disease. And now here the story starts. So there are so many molecular and biochemical, these studies have been done on this ashwagandha or Withania somnifera, but I just try to elaborate this particular thing, and that's why my topic of my this talk was the newer insight into the mode of action, anti-cancer mode of action of the Withania somnifera. So here I started just to, because if you target a particular cancer, if you deliver a drug in your preclinical model or clinical individuals, so these are the three types of cells generally the drug used to go and interact with. First is the normal cell, in that tissue in movement. Second is the whatever the cancer cell is there, whatever the organ you are targeting, and third is the small population present in that particular tumor, that is known as the breast cancer cell. So here I just put three different origin of cell lines and just tested the effect of Withapherin A at the microRNA level. Now for that we just MCF10 cell lines. This is the normal cells. The in vitro model was the MCF10 cell lines, and for triple negative breast cancer cells we use the MD-MB231 cells, and for the breast cancer stem cells we used MCF7 cells and just produced the breast cancer stem-like cells as per standard protocol. Now these are the three studies we have just sequenced the MCF10 treated with the same concentration, and that was the 2 micromolar. Okay, and we just submitted the data, and which will be publicly available after we just publish the data. And first study was related to the triple negative breast cancer study. Second deals with the breast cancer stem-like cells, and study third was the Withapherin breast cell, that is the breast normal cells. So I will be talking about the study 1, that is what the effect of Withapherin on microRNA expression modulation of Withapherin in triple negative breast cancer cells. So this was the basic experiment. That is what we studied, the effect of Withapherin A on two different triple negative breast cancer cells, MD-MB231 and MD-MB453, and we got these IC50s that was near about the 2 micromolar, and it is well established in the literature. Across the literature you will find near about 2 micromolar IC50 of this particular drug against various cancer cell lines. And this is what the entire sequencing data I summarized in a single slide, and here we found what the 413 differentially and significantly expressed miRNAs, and this is the B figure that shows the log fold change and the significance of the miRNAs across these particular sequencing data. And the C, here I put 20 most upregulated and downregulated miRNAs. So, and this is what whatever the miRNAs we got, the enrichment of target gene prediction was done, and the enrichment of those genes in the different, you can say, the gene ontology terms as well as the cake pathway analysis, what was done. And the beauty of this particular thing, we found that yes, in all these things, all the signaling pathways or the gene ontology terms that are related to the breast cancer or aggressiveness or metastasis, these genes, the target genes, were enriched on those geo terminology as well as cake pathways. So this slide shows you the validation of whatever the miRNAs we got in sequence. So we just picked up the 5 up and 5 down regulated miRNAs and validated using the qRT-PCR analysis. And out of 10, we just got the validation of only 7 miRNAs, out of which the first here you can say 181C5P. So that was the most upregulated miRNA in that particular picture. So again, we thought that whether this particular miRNA is differentially expressed in the normal cell versus cancer cell. So that's why here we have the MCF-NA, that is the normal cell line. The expression of this miRNA was checked in this year and compared with the triple negative breast cancer cell line, and we got that the down regulation of this particular miRNA was in triple negative breast cancer. So after that, this is the basic experiment that we just transfected these cells to study the functional aspect of this particular miRNA in the given in vitro model. So we just transfected the triple negative breast cancer cell with the help of a miRNA-1C5P mimic, and we got that yes, the experiment was successful because we were getting the miRNA expression in time dependent manner. In the last graph you can see here, and from here the study starts. Here we just co-treated the miRNA mimic plus the different concentration of Visaferin A, and it shows that here the WA30 denotes to WA30 and IC50. So here you can see just co-treatment of mimic with the phytochemical, that is here the Visaferin A. The expression of that particular miRNA was very high. Now this is the toxicity, time-dependent toxicity of the miRNA 181C. Here you can see the 24 hour, 48 hour and 72 hours in two different cell lines. A is MD231 and B is 453. Here you can see that the effect of the cell viability was decreased in time-dependent manner. Now, the mechanistic aspect of this particular mode of action, I just picked the two cancer hallmarks. That one was the cell cycle and second was the apoptosis. Okay, so here I just picked two of the cell cycle markers, that is CDK4 and cyclin D1, and these two protein markers are responsible for the transition of G1 to S phase of the cell cycle. So first A or B graph here you can see this shows only the effect of Visaferin at different concentration, that is at 30 and 50 IC50 concentration, and when you just mix or co-treated with the mimic plus various concentration of Visaferin A, you can see the drastic decrease in these two markers, that is the CDK4 and cyclin D1 in this particular graph. So it shows that yes, WA by working at miRNA level by up expressing the levels of the MID181C, and thereby the decreasing or arresting the cell. Again, this is the same experiment at protein level. Here you can see the decrease levels of the CDK4 and cyclin D1, and the decrease extent was more when you just make or the co-treat with the miRNA mimic as well as the different concentration of Visaferin A. Now this picture shows the apoptosis. Okay, so here this Weber dye, that is for the horse dye, the cells were just stained with this particular dye, and the cells were treated with a different concentration of Ethanoate somnifera alone as well in combination with this miRNA mimic. So here in the last slide you can see the mimic plus IC50 concentration, the extent of the apoptosis was greater. So in this B picture here, this shows the decrease in mitochondrial membrane potential, and the green cell shows the dead cell and the live cells in the green cell. So here you can see the extent of decrease in mitochondrial membrane potential. Now again, this is a very not much popular protocol, and this protocol has been utilized for the last 2-3 years or 5 years you can say. So it is for the semi-qualitative measurement of the apoptotic population of apoptotic populations. So in this particular thing, what do you used to do? The instrument is different. Flow cytometer, you used to do the same dye, PI and XNV, but this type of experiment you can do in the confocal microscopy. So here again we are getting what the live cells and dead cells, and these are all in the shade of the different concentration of the co-treatment as well as alone. Now this picture shows these generation of reactive oxygen species in the cells, because reactive oxygen species are known as the inducers of apoptosis as well as the toxicity of the particular agent. So this is again anti-cancer mode of action. So here you can see in WIC30 plus mirror mimic, you can see the loss generation was there. But an interesting thing was that we observed in the last thing, that is what we are getting the IC50 plus mirror mimic. You can see the disintegration of the morphology of the cells, and it shows that this particular combination is highly toxic, and the cells, they just shed off the integrity, you can say. So it shows the potency of this particular treatment. Now we started the effect of this particular combination or alone for the apoptotic markers, and this first graph shows the effect of the alone treatment or the co-treatment on the PARP expression, and second was the BCL2, and these are what the apoptosis related markers. Now third one is again the caspase 3 and caspase 8 related activities. Again you can see the very huge difference between the alone treatment as well as the treatment at the various concentration of isoflurane A, as well as in co-treatment with the mimic. Here M stands for the mimic and MC negative control, and WA30 and 50 are the various concentrations of the isoflurane A. Now this is again
please write this over.
Okay, sir. So this particular slide shows the caspase activity, caspase 3 and 7. This is the enzymatic activity done by the luminometer, and this is the effect of WA alone or in combination on the various, you can say, protein markers of the apoptotic. This is the thing, and this is the conclusion slide, and this is what the thing that has been done for the last 2-3 years. And this shows that yes, if you just treat with the cancer cells, with aflurane A, it upregulates the MIRN 181C FP, and it affects at least 2 of the hallmarks of this cancer, that is the cellular proliferation, and it is related to the down expression of CDK4 and CC and D1. And second is the 2 of the intrinsic mechanism, that is what the mitochondrial mediated apoptotic mechanism, and second one is trans-ligand and this particular complex mechanism. Here all are just convergent to the apoptotic signals. So this is, and that was related to the triple negative breast cancer cells, and this is what the another study, that is what the isoflurane mediated change of the mRNA expression in breast cancer is done by Namusfer. So that we got published few months back. Thank you.
Any questions?
So you have said that triple negative breast cancer is very aggressive cancer, and it generally occurs in early ages, and so what could be those possible reasons why this is actually growing more in India?
Okay.
I mean, there could be some more specific, because you have shown in comparison to US.
Yes. So that was a.
And what is that total percentage which you are actually observing in India?
So, 2 different questions and the answers are very much different. I am not able to speak on the exact answer of these particular 2 questions, because if you talk about the national registry program, and that is what related to the identification of number of the patients of particular cancer in our country, it is very dangerous, you can say, because in the last month there was a standing committee of the Rajya Sabha. They just said that this particular disease should be a notifiable disease, because it shows that we are lacking of the actual number of cases and actual number of, because we are living in a particular country where so much diversity is there, so much food habit is there, and so these all types are related to the cancer, because cancer is a lifestyle disease. So if you don't know the exact number of the disease, so how can you correlate with the geographical region as well as this type of thing? So for that reason I am not able to answer exactly.
But it is somehow, because in my studies also I was collecting samples from many sites of the.
So, excuse me, can I interrupt this? You can have discussions during the
Okay, thank you.
In this
Thank you.
Thank you. So the next speaker.